Hi there, my name is Blake Clark. I am an oceanographer in the Ocean Ecology Laboratory at Goddard Space Flight Center, and today I'm going to talk a little bit about the state of the art of linking optics and biogeochemical models to better quantify marine carbon cycling. And I want to give a shout out to my employer, USRA Guest Star, as well as the NASA postdoctoral program, of which I just finished up in April, and also OCB for hosting this workshop, which has been great so far, and I'm sure will continue to be so. And also to Maria, Seth, and Amy for convening this session. Also, I want to give a big shout out to the International Ocean Color Coordinating Group, report number 19, and all of the contributors, which are listed on the right. This report, which was published last year, was extremely helpful in sort of framing my thinking around the synergy between ocean color and biogeochemical/ecosystem models. And these reports are all publicly available on their website, and I highly recommend for both the experts as well as the novice users as well. So the first question I want to ask is, what are biogeochemical models? Because that word, biogeochemical model, those words can mean, different things to different folks. And when I started to put this talk together and think about what I was going to talk about, I wanted to do the Homer into the hedges meme, which is sort of just disappear because of the question of, the linking and the synthesis of optics and biogeochemical models is so broad and so big. But after thinking about it for a while and then reading the wonderful IOCCG report and sort of digging back into the most recent literature, I realized, well, this is actually a great topic and one where I can, teach a little bit to the folks who might not have a really strong background, especially coming from the modeling aspect, which is where I'm coming from. So broadly defined, biogeochemical models are a set of differential equations that are used to simulate some aspects of biogeochemical cycling using computers and numerical integration. And this is a definition that's very nicely laid out in the previously mentioned IOCCG report. And today what I'm going to do for the first part of this talk is use the NASA Ocean Biological Model, or NOBM, and then a couple other systems to show how modern biogeochemical models are representing these very sophisticated ecosystem properties. So the NASA Ocean Biological Model, or NOBM, is a part of the Global Modeling and Assimilation Office. It's primarily was developed by Watson Gregg and is now run by Cecile Rousseau, whose contact information is still on the left. And this is a very sophisticated model that predicts not only the flow and the physical transport of water in the ocean, but also things such as the radiative transfer of irradiance into the ocean, as well as the radiance back out of the ocean. And it includes many sophisticated biogeochemical processes, such as 4 phytoplankton groups, the multiple nutrient limitations, primary production, as well as the air-sea carbon dioxide exchange. So a very sophisticated model, and we could break it down even further. So this is sort of the highest level view, and look specifically at some of the types of things that are being simulated. So here I'm showing the biological portion of the model, and the radiative transfer portion of the model. And so you'll see that within each of these larger pools that there's many flows that are going on. And we could break it down even further, specifically at the radiative transfer model, which I'm showing here in this schematic. So this is a very interesting system because it actually predicts not only the downwelling radiance, hyperspectral irradiance, into the ocean from the atmosphere, but also the water leaving radiance, which can be directly compared to what a satellite would see overhead. So the model can predict the actual remote sensing reflectance through time. And just to break down one step further, these are the types of differential equations I'm talking about. We have the change in a property over time. Here, the 3 types of radiance and irradiance that are being propagated into and out of the water column. And so this is a very sophisticated model that has, almost a decade and a half of development behind it, if not more. And it's within a larger framework, like I said, at GMAO. And, a lot of time has gone into the conceptualization and the actual coding of this system and testing for that matter. So the NOBM is not the only model of its kind. There's numerous types of global ocean models that are used to predict not only the past biogeochemical cycling throughout the world's oceans, but also potentially project into the future, such as the Echo-Darwin model, shown here on the upper figures, with a recent publication on the left. And both of these models that I'm showing here assimilate satellite data directly to get a better prediction of the ocean color as well as the biogeochemical properties. And they could be used to relate back the in-water optical field, such as the remote sensing reflectance, back to what actual processes are driving those changes through time and space. And I'm going to come back to looking specifically at these global ocean models towards the end of the talk when I talk more about data assimilation. We also have models that are used to run and simulate processes on regional, local, and even daily scales. So here I'm showing the Doppio model, which is from the Mid-Atlantic, which stretches from south of Cape Hatteras north through the Gulf of Maine, as well as a forecasting system, which there's a session on ecological forecasting in the workshop this year that has been developed by a group out of VIMS that can actually predict the future dissolved oxygen forecast for Chesapeake Bay. And then here's another model that I worked on as a PhD student that is simulating very, very fine scale processes on the order of tens of meters and on the order of 15 minutes to better understand the linkages between the land and the ocean in Chesapeake Bay through tidal wetlands. So these are the types of scales that we're simulating. But also biochemical models can be used to just make really cool movies. So this is a system that I've developed as a postdoc where we're actually simulating the flow and transport of material from the land into the ocean at the Yukon River outflow. So nothing really to take home here quite yet, just that, if anything, we can use these biogeochemical models to make really cool movies for talks such as this. So now I'm going to get into sort of the meat of the talk, now that we've done our homework a little bit. And first I'm going to go through a case study where I, as well as some co-authors, worked on the conceptualization, parameterization, and validation, a lot of Asians there, of new model pathways, specifically looking at the photochemical degradation of colored dissolved organic matter, simulating processes that have been observed in the lab and then out into the coastal ocean. And then towards the end of the talk, I'll talk more about assimilating and validating ocean color into biogeochemical models. And then at the very end, I'm going to touch on sort of this concept of, well, what are the positive feedbacks between remote sensing and biogeochemical models? And how can we move forward in linking these two very useful tools? So I'm going to spend the first part of this section talking about how CDOM absorption and DOC molecular properties are directly linked, and how we can take the concepts that have been distilled in this paper by John Helms, for example, and put them actually into a numerical conceptual framework, and then code it from recreating what happened in the laboratory experiments and then scaling it up to a large coastal system such as the Yukon River. So the first idea is that for some background here, is that irradiation and distance from land are linked to changes in the molecular weight of DOC, which is due to photochemical degradation. And these can also be linked back to optics, where you can take the slopes of two regions of a CDOM absorption spectra and take the ratio of those, and those pretty well correlate to how much low molecular weight or high molecular weight DOM is in these systems. So these concepts, linking optics to molecular weight as well as processing from land into the ocean, can be used to construct a numerical model, a novel numerical model, which I did with some of my co-authors back in 2019. So we take this idea and bear in mind that DOC is obviously made up of thousands of types of compounds, and we could distill it down to simulate the breakdown of high molecular weight DOM into lower molecular weight DOM, as well as sort of this middle molecular weight DOM pool. And this paper was published in Marine Chemistry back in 2019. And we can test this by actually recreating laboratory experiments that were conducted in Chesapeake Bay. And we do this by essentially recreating the actual laboratory environment in the computer world, such as the amount of irradiance, the time of irradiance, as well as trying to parameterize the model to recreate what's shown in terms of the observations from the lab. And that's shown here, where we were able to pretty accurately recreate the breakdown of DOM from initial to final, as well as the time breakdown of CDOM over a period of 7 days. So we can take this idea from a very simplified system where we're just recreating laboratory experiments, scale it up to a much larger system, of course, by reparameterizing some important aspects of the model itself. So here I'm just going to touch briefly on actually simulating the CDOM absorption and the change in CDOM absorption due to photochemistry in a very large coastal system such as the Yukon River. So I'm not going to go into too much detail on this slide, but just to show that we're actually trying to represent the changes in the optical properties as well as biogeochemical cycling in the water column in space and time as the river discharge peaks upon ice breakup in the spring and then through the summer and into the fall. And we actually simulate 6 types of DOC, 3 of which can undergo photochemical degradation. And these are parameterized using in-situ data that were collected in these systems, which is key to having any good modeling system. And it's becoming increasingly more aware that in these sort of faraway places, such as the Yukon River, we need to go out and collect as much information, whether that's through remote methods or actually sending folks up there to go sample as possible. So the question is, well, what happens when there is no photochemical degradation? Another way to put this is, well, if we take that aspect of ecosystem cycling away through a what's called a scenario, then we can actually get a better sense of how important that is, both directly and indirectly, to carbon cycling in these coastal systems. And so, we can just turn the lights off and find out. Well, in reality, we don't want to turn the lights off because that's going to affect other things, specifically phytoplankton growth. So we can just set the parameters for photochemical degradation to 0. So the easiest way to see how important a specific type of reaction or series of reactions is to the cycling of nutrients and carbon in these biogeochemical models is to just turn a specific process off and then take the difference between the model run with that thing and the model run without that thing. Here showing what happens when we actually make photochemical reactivity of DOC 0. So what we're showing here on the left is the CDOM absorption at 443 nanometers. And so just as we would expect, when we have photochemistry turned off, you get a greater accumulation of CDOM in the surface ocean. So here we're taking photochemistry off minus photochemistry on, or the baseline scenario. And with these models, what makes them extremely useful is we can look at these things not only sort of in 2 dimensions, but also in 3 dimensions. Here I'm showing the difference in CDOM absorption along this transect on the left, coming out of the Yukon River plume into the coastal ocean. So just as we would expect, the CDOM at the outflow of the river, when you have photochemistry turned off, is elevated relative to the baseline scenario. But we could also look at other things through the ecosystem, such as the photosynthetically available radiation and the spatial patterns and how that relates back to what's occurring with CDOM. So interestingly here for this case, we have more photosynthetically active radiation further out into the ocean downstream from the river. So you sort of have the impact of this increased amount of CDOM affects the light field, the underwater light field, the further you get away from the river plume. So a little bit counterintuitive. And the reason that is because the near field is dominated by particles, which are not affected in this modeling system by photochemistry. So by having still the same amount of particles generally that are occurring in the near field close to the river plume, that's really dominating the attenuation of light through the water column. So it's these types of attribution studies where you can look at multiple aspects of an ecosystem in these scenarios and get a better sense of, in this case, how photochemistry affects the actual optical field in the water column and presumably phytoplankton growth. So that was just one example of how we can take these relatively widespread and unified concepts, such as photochemical degradation of DOM, and systematically scale them and include them into a numerical modeling framework beginning by recreating the observations actually in the laboratory, and eventually putting them into the ecosystem framework in these coastal, regional, or even global environments. So now I'm going to shift gears a little bit, and perhaps a little bit abruptly, and talk about the assimilation and validation using ocean color products in biogeochemical models. And we'll look at two systems, one regional around the North Atlantic, and then one global, coming back to the NASA ocean biological model. So assimilation of satellite data has been going on for quite a long time. And I kept coming back to this question, which is, which maybe somebody can answer in the comments, which is, how many ocean biogeochemical models are there? I don't know if anybody has that answer, but it'd be fascinating to sort of figure that out. But the two I'm going to talk specifically about in terms of data assimilation, one is this model by Goddard et al., where they actually assimilate the phytoplankton functional types into the model. So they're taking predictions from remote sensing reflectance from space and saying, by using a cost function, they are assimilating that information into the predictions of the model. And then the NASA ocean biological model, which assimilates the chlorophyll concentration observed from space, as well as other inherent optical properties such as the detrital absorption, the DOC concentration, as well as the particulate inorganic carbon concentration, to get a better prediction of the spatial fields through time of this modeling system. And what's really cool about the NOBM is that it's recently been used to actually recreate hyperspectral normalized water leaving irradiance throughout the world's oceans. So it can be a valuable tool to actually get a better sense of what's possible as we move into the hyperspectral age. And briefly touching on this model from the North Atlantic, where we can see that by assimilating these data products, here showing phytoplankton functional types, into the surface ocean predictions, we can get a better representation of properties such as the phytoplankton community structure. And secondarily, we can get a substantial improvement in things such as carbon dynamics, or here I'm showing the surface ocean partial pressure of CO2, as well as the net community production, or sort of the net carbon dioxide flux across the surface. So that just shows the potential power of assimilating these higher level satellite products, which are derived from remote sensing reflectance, into these systems. And presumably, as we move from the multispectral into the hyperspectral age with PACE and with Glimmer and with SPG, for example, we will be able to better distinguish these phytoplankton functional groups using the remote sensing reflectance information, and presumably then be able to better include them and assimilate them into the ocean model predictions going forward. So that's one example of a regional model that's assimilating one type of derived ocean color product, which is the phytoplankton functional types. The other example I wanted to touch on today, and like I said, there's numerous assimilative data assimilative models, but really only had time to briefly touch on a couple. But yeah, the NOBM, which takes in ocean color products such as chlorophyll concentration, as well as the inherent optical properties, here showing CDOM absorption, and actually assimilate that information into the surface ocean, and then recreate the IOPs through time and space, which can then directly be compared back to what's predicted from the satellites, as well as you can predict things that are not quite yet possible to observe from space, such as the recent study where they looked at the hyperspectral remote sensing reflectance shown here at 12 wavelengths, but actually going from the UV all the way through the red at really any wavelength interval that the user would be interested in. So this is kind of where the potential of these satellite data sets to both positively feedback into these models, as well as using the models to better understand the spatial variability that can be observed from space and the spectral variability that can be observed from space, how the two things can sort of interact. And the last thing I wanted to touch on using the NOBM is that by assimilating all this data, you can actually glean very, very useful and insightful information. Here showing the global ocean primary production over the last 2 decades. So by better representing the surface chlorophyll concentration of the surface phytoplankton community concentrations, then we can presumably get a better representation of things such as primary production or other things such as export, which Yvonne talked about. So it's these types of the ability of the models to incorporate satellite information, but also incorporate the uncertainty associated with satellite information. That's really where the community is moving towards, and then eventually potentially using the biogeochemical models to inform what these satellites are seeing from outer space. Which leads into my last few slides, which is conceptualizing my thinking and hopefully spurring some thoughts in the community and the folks who are watching this, and how can we use remote sensing and biogeochemical models to positively reinforce the aspects that they both excel at, to eventually potentially link ocean color, apparent optical properties, inherent optical properties, and processes with what's being predicted in these modeling systems. So here's a flow diagram that I came up with that was that I'm using to sort of think about how we can use these two different tools, remote sensing as well as in-situ sensing of optical properties, and combining them with what models predict, which are very sophisticated cycling and rates in time and space, to converge on something that that tells us about the optical properties of the ocean, as well as the optically active concentration of things in the ocean, where we can converge on something that is representative of the truth. So this is set up to so that these two sort of flows can kind of mirror each other, where eventually what we end up with is the derived IOPs as well as the derived spectral absorption for these different constituents, and how the concentration and rates are utilized in the processing up from just initializing a model field or getting the level 2 ocean color product from space, eventually being able to derive these rates and IOPs in the ocean. And lastly, what I really wanted to illustrate here was that because these two tools can simulate very similar aspects of the ocean optical properties as well as the biogeochemical cycling, with remote sensing reflectance using algorithms, for instance, as well with modeling biogeochemical models, calculating the differential equations through time and space, that we can use them to converge somewhere on the truth of what is actually in the ocean and what types of processes are occurring in the ocean at any given time. Now, this is very simplified, and I definitely have some thoughts on once it comes to the in-person session on the 22nd. But I just wanted to throw this out there, that really I think that there's a synergy as these biogeochemical models become more sophisticated and we move into the hyperspectral remote sensing world, where we're predicting the same thing from two completely different sources, and that should give us the ability to converge somewhere onto the truth, especially when we start incorporating things such as the biogeochemical Argo fleet, which will give us optics as well as concentration in the water column, and then other platforms such as moorings, suborbital missions, exports, for instance, which went out and measured so many things related to the carbon's flux from the surface ocean into deep, where by incorporating the in-situ observations with modeling capability as well as the ocean color products, we will be able to converge somewhere onto the truth of what's in the ocean and what's occurring in the ocean at any given time. Now, I'm going to leave it at that, and I would highly recommend if you guys are interested, to go check out the Biogeochemical Argo workshop, where they have all these wonderful talks that were recorded over the month of May. And so with that, and I apologize, I went a little bit over time, but I guess that's the benefit of having recorded talks. I want to thank all these folks who I've worked with, even going back to my PhD days, as well as my funding sources, which has been NASA, the NASA postdoctoral program, a USRA guest star, who I work for now, as well as the NASA Center for Climate Simulation. And so with that, I'm going to leave it with these 3 questions, general questions that I came up with in the process of making this talk. And leave my charge to the audience to come up with some questions of your own so that we can have a good discussion on June 22nd. It'd be great to hear from the community on what the experts think about, the future into the hyperspectral era of what's possible with incorporating optics and remote sensing with biogeochemical models going forward. So with that, I'll say thank you to everybody for watching, and I look forward to seeing you on June 22nd. Thank you.